and thank you everyone for tuning in. As Nikolai mentioned, I'll be presenting the fundamentals of Evosep One and its applications. Evosep aims to make proteomic sample preparation and separation for MS analysis 10 times faster and 100 times more robust. And it accomplishes that through using of ease of use, robust and reliable chromatographic solution that is built for high performance and to help standardize proteomic workflows. And it all starts with the cornerstone of our technology, the Evotip Pure. It is a offline desalting device that can be seamlessly integrated into proteomic sample prep workflows. Uniquely, it is integrated into the chromatography, where it is used as a single-use disposable trap column. The other half of our technology is the Evosep One. It's an LC that is built to deliver long-term reproducible chromatography. And it accomplishes that by one, using the Evotip Pure as a single-use disposable trap column, and using a partial elution to elute your analytes from the Evotip so that the highly hydrophobic material remains on the Evotip and is discarded and does not enter the instrumentation. Secondly, its unique pump box design allows you to have much shorter overhead times than traditional LCs, as well as it mitigates the stress on the mechanical components to minimize the wear and tear on them. Shown here is a cartoon of a traditional proteomic workflow where after your proteins are digested, right, the peptides are then desalted on some sort of desalting cartridge that require you to elute your analytes from the desalting cartridge into some sort of plastic container. Which then you dry your samples down, and when you're ready for LC-MS analysis, you resuspend the samples and transfer them into an autosampler vial. With Evosep's technology, those digested peptides are loaded onto the Evotip, right, and they are immobilized on the matrix of the Evotip and allowed to be washed. Once the analytes have been washed, right, they are actually ready for LC-MS analysis, where the Evosep One will elute your analytes from the Evotip prior to analytical separation using a partial elution, as I briefly described earlier. The Evosep One utilizes fixed methods that have been designed and optimized for it, as well as proteomic applications. And we have two buckets. We have our standard methods, which are used for most proteomic applications, and they are different based on active gradient length, and they are labeled based on the how many samples per day you could run based on that method. So for example, the 500 samples per day, that is our fastest method, and it has an active gradient length of 2.2 minutes. On the other side, the 30 samples per day, there is an active gradient length of 44 minutes. The specialized methods, these are methods that have been designed and optimized for specific applications. So starting from the left, the first four here, these are our whisper zoom methods, which they have been designed and optimized for high sensitivity applications, right? And they differ based on gradient length from 10 minutes all the way to 68 minutes in terms of active gradient length. And then next we have our extended method, where this method was designed for deep proteome coverage due to the fact that it is our longest method and has an active gradient length of 88 minutes. And then finally, we have an organic method, high organic method, which is used to analyze highly hydrophobic analytes. Again, these methods have been designed and optimized to ensure that they are plug and play and give out of the box consistent chromatographic performance. And you could expect that if you use the specified columns that we recommend. We have two lines of columns. We have an endurance line, which are columns meant to be ran at room temperature. And we have a performance line where the columns are meant to be ran at 40°C using a column oven. The Evosep One is controlled by Evosep One driver that could be installed and integrated into the mass spec software. And then finally, the Evosep One could be coupled to the major vendors' mass specs through the usage of optimized emitters in zero dead volume connectors. And again, the Evosep One can be controlled by a driver that is integrated and installed into the native mass spec software. And moving forward, I'm going to be asking some rhetorical questions that some of you may asked yourselves in the lab and showcase how Evosep's technology can answer those questions. To begin, I'm going to start with, what if your samples were running all the time? Evosep's technology allows you and enables you to run samples all the time due to its key features, starting with the Evotip Pure, because it is integrated into the chromatography and used as a high efficiency, single-use disposable trap column. And then the Evosep One uses a partial elution to elute your analytes and leave behind the highly hydrophobic material on the Evotip, which then gets discarded. So therefore, it is LCMS fail-safe and also truly minimizes sample carryover. Uniquely, the Evotip Pure can be used as a storage device where the analytes, once are mobilized on the matrix of the Evotip and washed, they can be stored at room temperature up to a week and at 4C for up to a month with minimal sample loss. The Evotip Pure has been designed and optimized to minimize sample loss throughout the loading process and storage, as I just mentioned, so you could rest assured that you could clean up and concentrate your sample even at lower inputs. And again, it could be seamlessly integrated into proteomic sample prep workflows. Another feature that enables the Evosep technology to run samples, to allow you to run samples all the time, is due to its unique design of the pump box, where it is designed to mitigate stress on the mechanical components so that you minimize wear and tear on them. Another key feature, or another reason why it accomplishes that, is through the usage of low pressure pumps. So they do the bulk of the work, where they elute the analytes from the Evotip using a partial elution, right? Form the analytical gradient, which will be used to separate your analytes on the analytical column, as well as the wash. And then we have a single high pressure pump that will isocratically pump those contents to the analytical column at different flow rates based on the method selected. And to prove that point, we ran an experiment where we loaded 5 Evotip boxes with different sample types and inputs. I'm only showing a subset of that data, but I will show the rest of it in the next few slides. So here, this is the data from a subset of samples that had 50 nanogram HeLa digest loaded onto the Evotips. And shown on the left is the pressure trace of those experiments, starting from run 1 all the way to run 403, right? And you can see that the pressure trace nicely overlaps. Looking into the chromatography of those subset of samples, we extracted 5 peptides across the gradient and looked at the retention times, right? And you could see that the retention times are reproducible across all 190 of those samples that were dispersed across all 5 Evotip boxes, and they had a standard deviation, excuse me, just under 2 seconds. The next question, what if you never had to worry about sample carryover? Going back to that experiment that I just described, again, we loaded 5 Evotip boxes with different sample types and inputs. Each Evotip box has 96 Evotips, which correspond with 96 samples. Digging into the mass spec data in terms of the sum precursor intensity, for box 1 here on the left, you could see that it is reproducible across different sample types and inputs, as well as I failed to mention that we also sprinkled in blanks throughout all 5 boxes. So focusing on the blanks in box 1 on the left here, you could see that the sun precursor intensity is negligible no matter following a batch of HeLa digest or plasma digest, right? Now focusing on the data on the right, which is the mass spec data from box 5, right? Here you could see that the sun precursor intensity was reproducible across different sample types and inputs, comparing it back to the samples from box 1. And importantly, looking at the blanks again, you could see that the sun precursor intensity is negligible no matter following a batch of HeLa digest or plasma digest. And this is a testament to Evosep's technology due to the fact that the Evotip Pure is used as a single-use disposable trap column, and the Evosep One use a partial elution to simply elute your analytes and leave the highly hydrophobic material behind to be discarded. My next question is, what if you could immediately improve your sensitivity? As we all know, that with lower flow rates, we significantly improve sensitivity, but we dramatically decrease the robustness and reliability of our instrumentation, right? But due to the fact that the Evosep One's pump box, the low pressure pumps do the bulk of the work, right? You could rest assured that you'll get the same robustness and reliability, no matter if you're using our fixed methods that use a flow rate of 4 microliters per minute, or our whisper methods, which use a flow rate of 200 nanoliters per minute. And to showcase that our technology could answer that question, we ran an experiment where we loaded Evotips with different sample types and inputs, and all the way down to single cell levels at 250 picogram. And we used our Whisper Zoom ADS-PD method, which again, these methods are designed for high sensitivity applications. You could see that the sun precursor intensity across the different sample types and inputs, right, is reproducible across the board. Further looking into the blanks that were ran after HeLa, a batch of HeLa digest or plasma digest, you could see that the sun precursor intensity were negligible. And to hammer this message home, we also ran a different experiment where we loaded roughly 340 Evotips with 250 picograms of HeLa digest and used our fastest Whisper Zoom method, the 120 samples per day, that has an active gradient length of 10 minutes. And looking at the retention times of 5 extracted peptides across the gradient, you could see that the retention times are reproducible across all 340 Evotips that were loaded with 250 picograms of HeLa digest and had a standard deviation of around 2 seconds. And finally, my last question is, what if it was easy to automate workflows? Evosep's technology can answer this question due to the sole fact that we have automated the Evotip loading protocol so that you can combine the automated Evotip loading protocol with different automated proteomic sample prep workflows, such as enrichments, fractionations, or just your typical sample cleanup, right? And due to the fact that the Evotip was designed and optimized to minimize sample loss throughout the loading process of your sample and storage of your sample on the Evotip, right? You could truly minimize your workflow so that you only process what will be loaded onto the Evotip. So you decrease the sample input so that you could have more replicates. You use less reagents and less costly enzymes to make the whole workflow more cost effective. The Evotip loading protocol is compatible with the OpenTrons OT2, the Agilent Assay Map Bravo, and soon we'll be releasing the workflows for the Beckman Coulter Biomech i5. These individual liquid handlers require specific Evotip loading adapters to use the workflows. The Evotip, the automated Evotip loading protocol relies on the liquid handler to mimic exactly what is done by the manual loading process with the same results, right? But here we found a clever way that we could actually have each of those steps inside the Evotip separated by predefined air gaps that could be pushed through the Evotip through using the positive pressure of the liquid handler. So here, as I mentioned, we mimic the manual loading process. So the bottom layer essentially equilibrates the matrix of the Evotip, right? The second layer is your sample that will be loaded onto the Evotip, and your analytes will be immobilized on the matrix. And then the top layer is a combination of your wash and also of your solvent that will be left over on the inside the Evotip to make sure the matrix does not dry out during storage or while your Evotips are sitting on the Evosep One deck waiting for LCMS analysis. So here in this video, you could see that the bottom layer is getting pushed through by the liquid handler and equilibrating the matrix, and then now your sample is being loaded. And your analyte's being immobilized on the matrix. The top layer is now being pushed through. So again, only a small portion of it will be used to wash your immobilized analytes on the matrix of the Evotip. We offer a few protocols, and these are simply just to inspire. This isn't the end all be all, right? As I mentioned, due to the fact that the automated Evotip loading protocol could be solely done on the liquid handler. You can combine it with other automated proteomic sample prep workflows, right? And the workflows that we offer rely on magnetic beads to capture your desired proteins, wash away the stringent buffers that were used during the lysis process or the resolidization process. And it's followed by on-bead digestion, where then the digested beads are then immediately loaded onto the Evotip. The three workflows are packed digestion workflow, which this is just your typical proteomic sample cleanup. And we have two plasma workflows, one for neat, and the third one, which we label as magnet. This is for to enrich for a subproteome of plasma associated with membrane-bound vesicles. Starting with the pack digestion workflow on the Agilent assay map for Bravo. Here, we allow you to choose different number of samples to be processed. Also choose different digestion times from 30 minutes to 16 hours. The digestion is happens on the deck of the assay map Bravo on a heating plate. And then we also allow you to designate how much of the digested peptides are loaded onto the Evotip based on percentage. And here, the digested peptides are immediately loaded onto the Evotip. So you start with your sample, start the protocol, and then you end with loaded Evotips that are ready for LCMS analysis. And to show the sensitivity of the workflow, we did a serial dilution experiment where we processed different amounts of sample throughout the workflow, starting at 5 nanograms all the way to 1,000 nanograms. And using our whisper zoom methods, again, these are designed for high sensitivity. We were able to get reproducible results in terms of precursor counts and protein group counts across all inputs, even down at 5 nanograms. Digging into the percent median CVs for the protein groups of the 10 nanogram samples, the 100 nanogram and 1,000 nanogram samples with both whisper zoom methods, you could see that the median CVs were all below 20%. Switching over to the plasma workflows, all this work that I'm going to show was done on the Opentron's OT2. right, due to the deck space and the workflow, right, you're able to process 192 samples, which is 2 plate worths of samples throughout the workflow simultaneously. Either it's neat or magnet, but we also give you the option to simultaneously process 96 samples through the neat plasma workflow as well as the magnet plasma workflow. And to showcase the reproducibility of the automated plasma workflows, we ran an experiment where we loaded plasma samples through the neat workflow and through the magnet workflow. And digging into the mass spec data in terms of precursor IDs and the protein group counts, you could see that we got reproducible results across all sample types, respective of the plasma workflow. Digging in deeper into the data, right? Let's focus into the plasma, the neat plasma data. Uniquely, we were able to detect the same subset of precursors and protein groups across all samples within the neat plasma workflow, right? Designated by the dashed line here. So we were able to detect the same 1,100 protein groups across all samples, and anything above the dotted line or dashed line is unique to that sample. We saw the same trend for the samples that went through the magnet workflow, where we were able to detect the same subset of precursors and protein groups across all samples within the samples that went through the magnet plasma workflow, designated by the dashed line. So we're able to detect around 400 protein groups in all of the samples that were the same protein groups. And anything above the dashed line is unique to that sample. Looking into the percent median CVs, you could see that for samples that went through both workflows, their median CVs for protein groups were around 10%. Now I'd like to take some time just to touch on our applications notes. They are well written, clean, concise, and have a plethora of nice data. And they all kind of build on one another, where it starts with the Evosep One methods, followed by the cornerstone of our technology, the Evotip Pure, and then some of the applications that benefit from our technology. And now, due to the fact that we have automated the Evotip loading protocol, now we have fully automated end-to-end solutions that some applications benefit from. And then lastly, we have a valuable toolbox, which is our website. Here, you could see old webinars, sign up for new webinars and events, surf the web shop, find more information on our products and methods. And more importantly, we have a resource tab that has a library that has most of the pre-reviewed papers that have been published, and you could filter through them based on application, mass specs used, as well as the Evosep One LC methods used. Thank you. Appreciate your time. Looking forward to the next talk and the Q&A session.